Yes, I'm very happy to be here and to be playing at home. And to be part of this wonderful symposium. So, like I was also reading Wiener's stuff when I was a PhD student, and when I got the invitation, and it also mentioned something about opinionated takes on cybernetics. So I was thinking about the different components of cybernetics, because I only have a few minutes. And I thought about these ideas of collective behavior and neural network that were also in Wiener's writing. So I thought I would focus on, try to be also a bit provocative and spur some discussion on how we can make sense of neural function using neural manifolds. And I want to provide some evidence to make the case based on work on invariances by several groups and what this may all mean to the organisms and us. So why neural manifolds? So, imagine that you're interested in understanding how this monkey's brain enacts his decision to grab a mango. So, the classic way to approach this is to essentially look at the independent activity of single neurons in the brain and their correlations with different variables of behavior. But, we know that many biophysical constraints, such as those arising from circuit connectivity, neuromodulators, common input, and so forth, make these neurons covert in specific ways. So our proposal, and that of other groups, is that brain function, neural processing, if you want, is based on these collective patterns of covariation. So to be a bit more formal, we can think of the brain as a system that has a very high dimensional space in which it accesses the activity of a neuron. And the activity of these neurons is not free to traverse due to these constraints the entirety of this space, but it remains confined to a low-dimensional surface. That is what we call a neural manifold. So what we are proposing is that when the monkey is grabbing this mango, what is essentially doing, what the brain is doing is using a behavior-specific manifold to enact this behavior. So in other words, the proposal I want to bring to again, spark some discussion is that these manifolds are doing the doing of the behavior. So how can we take this very loose theoretical framework and use it in our experiments to hopefully achieve some understanding? Imagine that we've recorded from a few neurons in the brain. So what we're going to do is look at their collective behavior by basically building a state space in which each axis is the activity of one neuron. And we're going to represent their collective activity that will form some trajectories that I'm going to loosely call latent dynamics. And now that we have these collective patterns of activity, we can use our favorite dimensionality reduction or manifold learning technique and try to estimate the neural manifold that underlies these trajectories. And our proposal is that when you are doing this, what you are doing is approximating this true biologically meaningful neural manifold that is doing the doing in the brain. Okay? So what are neurons in this view? So you can think of neurons as random projections or partial glimpses of what the manifold is doing. So you are sampling what is going on, and you may get more or less lucky and get information about what the manifold is doing, and you can get very lucky and learn a lot about what the manifold is. And I think this notion kind of demystifies a bit these ideas of mixed selectivity or changes representational drift and so forth, but this is not what I want to talk about today. What I want to mention this is because if you have enough views, then you know what is going on, right? If you sample from enough neurons, you can reconstruct this manifold. And of course, since I'm Spanish, I have to show a Picasso. So I've made 3 big claims in this introduction about manifolds that I want to address during my talk. The first one is that manifolds capture meaningful biological constraints. The second one is that manifolds may afford scientific understanding, so compressed understanding. And the third one is that neural manifolds may be doing the doing in the brain. And of course, this is ranked in how speculative these statements are. So, there's a good news for the community is that we have increasing evidence that manifolds reflect intrinsic constraints. Want to show 2 sets of studies. The first one focuses on invariances across behavioral states. So for example, the Fit and Perras group has shown that this circuit in the mouse thalamus that relates to head direction has basically a ring attractor manifold that activity moves along it and signals where the mouse is looking at. But the interesting thing is that when the mouse is asleep, regardless of whether it is REM or no REM sleep, this activity is still traversing this manifold, suggesting that there's a fundamental constraint on the circuit that forces it to be there, even when the animal is not behaving. And by the way, the same is true for the toroidal manifold that underpins great cell activity in mid and entorhinal cortex. So when the mouse moves around, the activity moves in this toroidal manifold, but it also remains confined to the manifold when the mouse is asleep. Okay. Again, suggesting of fundamental constraints on what the activity may do, that may be evolved. But there's another series of studies that provide evidence that manifolds capture fundamental constraints. And these are invariances across different ways to drive the system. Okay, so you can drive the system naturally, for example, by presenting it as stimulus, or artificially with optogenetics, and get the same collective behavior. So for example, the Dizeroth lab has shown this very strikingly. So they trained mice on a go, no-go task based on visual gradients. And they use calcium imaging to identify neurons that were very strongly tuned to the go or no-go stimulus. And then they could optogenetically activate these neurons with holography. And mice would do the task basically as well as they did with the stimuli, which is already quite cool and could be like a good philosophical discussion. But what matters for this talk is that they were also recording from many tens of neurons at the same time that they did this. And what they found is that the collective activity in the manifold of these recorded neurons were very similar when the mouse was responding to a visual stimulus or when they were driving with optogenetics these few neurons. Okay, again, suggesting that there's constraints that make these neurons work together in the same way across conditions. So now I think this is important because, I think many of us are interested in understanding. And what I mean about understanding is a bit in the Henk direct sense of it, of having intelligible concepts or objects that we can mentally operate with. So have things that we can use in our imagination, even to predict or have intuitions of outcomes of experiments and measurements. And one of the concerns in the community is whether the intuitions that we derive with manifolds are grounded in biology or not. But I would argue, based on the evidence I have presented and some other studies, that this understanding is indeed grounded in biology. It is just grounded at a level that is not the usual single neuron or neuromodulator level that we're used to. It's more an emergent level that captures basically the consequences of all these components working together. Okay? So let me give you a few examples of what these quote-unquote understanding or descriptions look like. So many of them are based on geometry. So one of them I already mentioned, this ring attractor manifold that underpins the activity of high direction cells in thalamus, that, by the way, has also been found in the zebrafish, an animal that we've learned a lot about thanks to the wonderful talk by Misha. And you can also have other examples. One set of studies that I'm sure everyone here has seen before, so apologies for that, is work by Mark Churchan with Krishna Shenoy, where they address this very puzzling observation that the same neurons in motor cortex that cause the movement are active before when the monkey prepares the movement, and they don't cause a movement, right? How come the same neurons are active in both conditions? So what Mark and Krishna and others showed with a really cool series of studies is that what is going on is that when the monkey is preparing a movement, the activity converges to an initial state. So one of these gray dots, this is actual data. And as the movement is unleashed, the dynamics evolve along low-fold trajectories. And the interesting insight here is that all these preparatory states live in one specific direction of the manifold, and the activity related to movement lives in an orthogonal direction to it. So you have two different functions performed by the same circuits just by exploiting two different directions, because you can move along the preparatory direction without having an effect on the movement direction. Okay? So this inspired a lot of work looking at orthogonal directions doing different things in the brain or correlating with different things in the brain. So for example, in mouse visual cortex, stimulus-driven responses and activity that relates to fidgets and arousal live on orthogonal directions. Also in hippocampus, we've known a long time about places. So this is basically a manifold that correlates with the position in a space. But if mice have to integrate evidence to make a decision as they run, then this evidence is accumulated in an orthogonal direction of the manifold. Okay, so this is one more example. And the last one is not about a population doing two different things, but a neural population doing something and talking with a downstream area. And this is work from Byron Yu and Christian Mahens, where they show this
communication directions that capture interactions between areas that are orthogonal to private function. Okay. And all these examples are based in geometry. Just to give you one example of another set of intuitions, this is work from Joe Patton's lab here, where they were looking at this interval estimation task in rats. These are recordings from striatum, and basically rats had to do time judgments. And what they found is that the activity always remains confined in this curvilinear manifold, and the speed along which the dynamics evolve along this manifold depends on the time that the rats are judging. Okay? So this is another kind of explanation, this time not based on geometry, but on dynamics. Okay? So, or description if you want. But the last point that I wanted to make, and this is the most provocative, is that perhaps manifolds may be doing the doing in the brain, or in other words, they may be meaningful to the organism. So this is why I use the word title and the word meaning on my title. So, and of course, this is a very speculative argument, so bear with me. So, we've known like forever that, or since the times of Darwin's grandfather, who apparently wrote sonnets about evolution through natural selection, but they were very bad, so no one cared. So we've known for this long time that basically animals, evolution through natural selection shapes the behavioral repertoire of animals. And what this means is that you acquire traits that you have to pass to your offspring, right? And this means that your brain has to be able to change in order to enact those behaviors. And you have to be able to pass the instructions to the developmental program to build brains that are able to enact these behaviors, right? So this was in the back of my mind since we got our dog Luna, and Luna is a Shiba Inu, is an ancient breed that hasn't been very crossbred, and Shibas have very stereotypical crazy behaviors that they all do. Just go on Instagram or whatever and you get millions of memes. And I was fascinated by how can you encode these complex behaviors through some genomic bottleneck and then basically generate another instance of this. Okay, and of course, we know there's not enough information to encode the details of the circuits, but the point that I want to make is that perhaps we have enough information to specify the overall statistics of the connectivity of these circuits to specify the manifolds that will mediate these behaviors. Okay. So let me give you a few examples of this, of how we get the same collective behavior regardless of the implementation by low-level components. So the first example comes from Eve Marder's, I think, very important work on the stomatogastric systems of crustaceans. So she has this 20-year-old paper where they were looking at these 3 neurons that form a central pattern generator that basically opens and closes the esophagus, if I remember correctly. And what Eve's lab realizes, and you can see the pattern of these 3 neurons, and they have a name. So they could culture these neurons and basically look at many circuits that if you look at the behavior of these 2 circuits, they look identical, right? But the beautiful thing is that they can also measure the intrinsic and extrinsic properties of these neurons, and you can see that these 2 sets of bars look remarkably different for both intrinsic and extrinsic properties. So you are getting the same overall collective behavior of this oscillator with very different implementations. Okay? And perhaps, this is, to the organism, this is fine, right? What is meaningful to the organism, quote unquote, is that it's able to digest the food. And this is, this is adaptive, and this can, this is how the organism can survive, right? By digesting this way. So this is a very simple example, right? It's a 3-neuron system, but we can scale in the spirit of the symposium things a bit. So we can scale up, and we can look at the almost entire brain of the C. elegans worm. And there's this really nice paper by Brennan and Project where they will basically, they started from this observation that you can take around 100 neurons and predict changes in behavior very well. And you can subsample these neurons to just 15, and you can still do very good predictions. But if you take a decoder trained in these 15 neurons and you apply it on a different animal that is genetically identical, the decoder sucks. Okay? So the relay and basically goes to the chance level. So the relationship between these 15 neurons, and again, this is a C. elegans, so these neurons have names, and we know a lot about them, or these people know a lot about them, and behavior has changed completely. The remarkable thing for my manifold manifesto that I'm kind of laying out today is that if you do dimensionality reduction, what you find in a worm is this beautiful manifold with two loops where the positioning along the manifold relates to behavioral events, and you can uncover this manifold in all the worms. And you can even leave worms out and basically predict what a different worm is doing. So this again suggests that there's multiple realizability of these collective dynamics in an entire brain. Although, of course, there's the caveat that we know that neuropeptides and other biological processes, and again, Misha introduced some of these aspects as well, may be important in the worm, right? But it's one higher order example. And we can scale things up more to mammalian cortex, and this is work done by Mostafa Safai in the lab, who is somewhere there, and Joanna Chang with Matt Perick and a few other collaborators, and this is kind of starting with the same idea that these behavioral adaptations of animals have to be somehow encoded in the brain, and that we know that monkey brains, imagine that those are monkey brains. I thought actual monkey brains before lunch would be gross. So that these adaptations, we know that these brains are unique in the cellular identity, but the way that these neurons are organized in microcircuits, layers, columns, regions, so forth, is preserved across animals, right? So you could think that maybe this preserved organization in the circuitry leads to these brains instantiating the same, we call it neural landscape, maybe we should have called it platonic manifold to be cheekier, of the monkey brain that allows monkeys to do monkey things, okay? So this is the hypothesis, it's very loose, but we can make it more specific by saying that if this is true, if we take two monkeys performing the same behavior, and they indeed instantiate this platonic manifold, if I record their activity and look at the collective dynamics in the manifold, they should be the same. Now, these two dynamics, I'm looking at them from different neurons, like I showed you with the Garnica example. So the only thing that we should need to do is just imagine that blue is my left hand and green is my right hand. We only need to look at the way to look at them to find it in order to show that they are the same, okay? So we're just doing a linear transformation. So let me show you an example. The paper is published, so you can have a look or talk with Mostafa during the break. So we started, one of the tasks we looked at were monkeys doing center outreaches. So you can see the movement of the hand for both, in both monkeys. They are very stereotyped because these monkeys were Olympics level of this task. These are neurons in motor cortex, so they look quite different. And we did dimensionality reduction, and these are the trajectories for these 8 reaches in the 2 monkeys. So you can see that at first look, they are quite different, right? But you can see that the organization of the colors is similar and matches that of the behavior that are the targets. So they're in this little clock thing. Okay. So now the moment of truth is, can I do this alignment left hand, right hand thing and show that these trajectories are the same? And they indeed are. And we can measure these correlations across all trials. The similarity of these trajectories based on correlations are extremely high, well above many lower bound controls that we did. But more importantly, very close to an upper bound control that was asking how similar are these trajectories within monkey, within neurons, within they. So we are doing very well compared to that. Not only in this example, but across hundreds of comparisons. And I should say that we could also use these trajectories to build a decoder in one monkey and predict how the monkey was another monkey was moving the hand point by point very well. Okay. And this is not only about receiving sensory feedback of the same movement that is being performed, right? Because we could also look at this preparatory activity that I described earlier that precedes movement, and we could align the activity even if the monkey was not moving at all, and decode the future executed movement that one monkey performed from the activity of a different monkey. Okay? Now, I basically started this argument saying that this should work, first based on behavioral similarity, and the paper has lots of examples where we look at decrease the behavioral similarity and this goes down, but also at having similar statistics in circuit connectivity, right? Which I propose or I speculated is what the genome may use to encode this. So, sadly, we didn't have data from neurodevelopmental models or anything that allow us to have the same behavior and different connectivity, so we had to build our own data. If you have the data, by the way, come find me during the meeting. I would love to talk about it. So what we have to do is engineer the data using recurrent neural networks that we trained on this monkey task, and then basically we added a parameter to the cost function that I'm going to call alpha that allow us to basically force networks to explore more the solution space, the connectivity space. So you can see this here. On the x-axis, you have alpha, and you can see that networks become more complex and the weight distributions larger as we increase alpha, okay? But the important thing is that networks with the same alpha still have very consistent behavior between them. So we have consistent behavior and much more varied connectivity, which is what we wanted, right? Because now we can test the idea that it's not only about behavioral similarity, the similarity in the underlying connectivity drives this preservation of dynamics. So this is exactly what we found. You can see that as I make these plots with increasing alpha, the similarity of the dynamics goes down. And I also want to connect this back to Eve Marder's results, because there what they found is that you can have many different instantiations, but all this same level, that is what low levels of alpha gives us. If you go very far beyond that, then you get very different solutions. And this is a point that Eve makes in her paper, that all these values that they found were within a very small range, thus combined in very different ways. So I want to finish scaling things up to go to something that is very primate, some may say only human, that is more cognition. So this allows us to look at tasks that are implemented by similar brains, but that can be solved in different ways. So we don't do this in the lab. This is a small project that started by Joanna Lazar, who is an amazing PhD student in the lab, who just joined. So basically what we were looking at is this publicly available data set of PFC recordings in monkeys while they were learning to associate different cues within session to different targets, and then they had to touch this target on a touch screen. So the typical decision-making task. And what we did was look at this delay period when there was no cue information or target information, and use decoding to try to infer whether the monkey was being more prospective, so thinking about the target, or retrospective, thinking about the cue. And with this, we could define a strategy space and measure basically the similarity in the strategy and see if it was related to the preservation of these dynamics, okay? And what we found is that if monkeys are adopting the same strategy, these dynamics are very preserved, but as the strategies become very different, so one monkey is thinking about the target and the other the cue, these dynamics are less preserved. And I don't want to say anything else about it, just come to Joanna's poster because she's presenting all this with a lot of detail today. And by the way, the coolest part is not here. So, this is all this that I've been talking about is based on linear methods, right? But I want to say that I think that these insights with very simple methods may actually, are only the tip of the iceberg. Like there's now a lot of work that we're going to hear about. For example, Eve Dyer's work on these foundation models using LLMs of neural statistics that show invariances across animals. Ayesha is going to tell us about wonderful work on models of dynamics across animals, and there's much more, as I said. So, I think this all relates to the idea that there are these invariances that may be meaningful to the organism. So perhaps this is what is, used by natural selection to encode behaviors that are useful and meaningful to the organism. And therefore, this is why I would argue it is meaningful to us to look at manifolds, because we can get compressed understanding of something that is grounded in biology. So let me summarize this short manifold manifesto. So I've tried to define neural manifolds not only as ways of describing data, but a way to capture collective patterns of behavior in neural activity that arise from meaningful biological constraints, and that can allow us, and I've given you some examples of these constraints, like invariances across behavioral states, across ways of driving the circuit. I've also made the point that they give us some compressed descriptions that may lead to understanding of these neural processes driving behavior. And I've given a few examples of this. Of course, there's a lot of work to be done. It would be great if we can look at these compressed objects and try to get what the algorithm that is driving the behavior is. This is on us to do, if it's possible at all. I've also, made some argument about manifolds maybe having causal power in the sense that this level seems to be preserved while there's multiple realizability of the implementation. So I've talked about crabs, or sorry, lobsters, worms, monkeys. And yeah, I just wanted to finish by thanking the organizers for putting this together and thinking of inviting me. My wonderful lab and the collaborators, everyone involved has contributed to the work I've presented today. Big shout out for Matt and Devika. We wrote a piece on manifolds that kind of is a bit of the foundation of today's talk. And also we have 3 posters in early stages, so please stop by to give these wonderful people feedback. And yeah, we are hiring, and as Guillaume said, we are hiring here. So thank you for your attention.